This is me Arju here and I welcome you to my channel Mr. Teeth, where your dental concept get dissected. So this is our part 2 video of the dentin, and in this video we will be covering with the different important topics of this chapter. So without delaying much, let's get started. So in this video we will be covering with all those 12 topics, okay, 11 topics. So our first topic is dentinal tubules. So in dentinal tubules, as I have written here, so it extends from the pulpal surface to the dentino-enamel junction or dentino-cementum junction, okay. So dentino-enamel junction or dentino-cementum junction and are perpendicular to the them, okay. So first I have different pictures of those, okay. So I have figures of those dentins, okay, dentinal tubules, how they are arranged in different parts of the tooth. So I will be showing those pictures later, but first we will deal with the theory, okay. Theory portion we will cover first, then after we will see the pictures. So actually the dentinal tubules are the tubular structure, okay. So just think like, okay, so just think this is a pen, okay. So this is a tube and inside this pen, okay, so inside this ball pen there is a refill. So this is a dentinal tubules, and inside this dentinal tubules there is a odontoblastic processes that is resembling that of a refill, okay. So just make concept like that, okay. So dentinal tubules are just a tubular structure that extends from the dentino-enamel junction or dentino-cementum junction to the pulpal surface, okay, and they are perpendicular to them. So they are perpendicular at both the sides, okay. So in dentino-enamel junction 2 and also in a pulpal side they are perpendicular. Similarly, in our another point, as I have written, odontoblastic processes runs in the canaliculi of the tubules. So odontoblastic processes, they are just resembling like that of a refill in a ball pen. So they runs in the canaliculi, okay. So there is a lumen in this tubule. So in that lumen, the odontoblastic process, okay. So this is a dentinal tubules, and here in the lumen of the dentinal tubule there is a presence of the odontoblastic process. And as I have discussed earlier, odontoblastic process is derived from the odontoblast, and odontoblast is present in the pulp, okay. So just watch that video too. And after that I have written represents the configuration that was taken by the odontoblast during the development. So this point is very important here. So as I have told, okay, I have not told it, okay, but then also as we know that dentinal tubules are arranged in S-shaped pattern, okay. So that is a primary curvature of the dental tubule, okay. If you have heard about those primary curvature or secondary curvature in your theory classes of your university, okay, then it's okay. If you not have heard those terms, okay, so I will be dealing with those primary and secondary curvature in this video, okay. So what I have written here is that it represents the configuration that was taken by the odontoblast during the development. So during the development, okay, I will be discussing with this in odontogenesis. So during development, when there is a development of both the ameloblast and the odontoblast, okay, firstly, so they at that time are closer to each other, okay, but after the deposition of the matrix, okay, and there happens the mineralization, so that odontoblast go towards the pulpal side and there is a creation of the dentin, okay. So what I mean to say is that, okay, during odontogenesis, just think this is a ameloblast, okay. Ameloblast means it gives the enamel, and odontoblast means it gives the dentin. So think this is a ameloblast and this is a odontoblast. So what happens is that, okay, so this odontoblast move towards the pulpal side, and here it is a pulp, okay. So there is this portion is a pulp, this portion is ameloblast portion that gives the enamel, okay. So it is giving enamel and this is giving the dentin. So what happens, this odontoblast, okay, so odontoblast moves towards the pulpal side, okay, and this releasing the matrix, okay. So it is forming the matrix upon which the mineralization is happening. So in that process what happens is that odontoblast move towards the pulpal side forming the matrix, and in those matrix there happens a mineralization and there is a formation of the dentinal tubules. So dentinal tubules and the dentin, so that this dentinal tubules represents the configuration that was taken by the odontoblast during the development, okay. So odontoblast take this type pattern, okay. So it moves in a S-shaped pattern, okay, towards the pulp. When it moves towards the pulp, okay, so it takes the S-shaped pattern, so it is easy to understand. After that, terminally tubules bifurcate at a 45 degree to form Y-shaped tubules called terminal tubules or the branches. So what I mean to say in this point is that, okay, so terminally when, okay, so I will show you one picture for this, okay. So here in this picture, okay, here in this picture, as you can see, okay, so in this picture, this black lines are the dentinal tubules. Okay, so they are the dentinal tubules that are running from the pulp towards the enamel side. Okay, so in this figure, as we can see here, this is a termination of the dentinal tubules at the dentino-enamel junction. So at their terminal ends, okay, the dentinal tubules give the extension. Okay, they bifurcate. They are resembling like a letter Y. Okay, so they are resembling like a letter Y. So they are bifurcating at the terminal end, and those branches, okay, those branches that are formed by the termination, okay, by the bifurcation, so those branches are called the terminal branches. Okay, so they are the terminal branches of the dentinal tubules. So same I have written here that, okay, so terminal tubules bifurcate at the 45-degree to form the Y-shaped tubules called terminal tubules or the branches. After that, I have written here terminal tubules are more pronounced in a root dentin than the coronal dentin. So you should remember this. This is important. Okay, so in radicular dentin, radicular root dentin and coronal dentin means okay, root dentin means that dentin that is present in the root. Okay, root of the tooth. Okay, and coronal dentin is that portion of the dentin that is present in the crown of the tooth. Okay, so now in next point, as they are written, okay, so it is S-shaped or doubly curved, that is a primary curvature of this dentinal tubules, and the first curvature is directed towards the root, another is towards the crown. Okay, straight course at the root and the incisor and cuspal areas. Okay, so it is a root apex. Okay, so root apex is it. So I will show you the pictures. Okay, so in pictures I will explain this clearly. So in these four pictures, okay, so in these four pictures, as we can see here, okay, so in those pictures, okay, we can see that this green line, okay, so this green line is a dentinal tubules. This green line is a dentinal tubules. This is a pulpal side, this is a pulpal side, pulp side, and this is enamel. Okay, and this is cementum here. Similarly, this is enamel here, and this is a cementum here. Okay, so this picture explain us different things. Okay, we can clear our concept. We can clear our main concept by those pictures. Okay, so in first picture I will explain this picture. What is happening here is that, okay, so dentinal tubules, okay, that is present in the dentin, so it runs from the pulpal side towards the enamel side. Okay, and the thing that we can understand by this curvature, okay, by drawing the dentinal tubule like this is that, okay, the significance is that, and the drawing of the dentinal tubules in this root portion, radicular dentin. So this is a coronal dentin. Okay, so this is a coronal dentin, and this is a radicular dentin. Okay, this is a radicular dentin. So the significance of drawing dentinal tubules in this pattern in coronal dentin, and the significance of drawing this dental tubules in radicular dentin is that, okay, so in coronal dentin, the curvatures, okay, so the curvatures of this dentinal tubule is more, but in the radicular dentin the curvature of this dentinal tubule is less. Okay, so I have drawn like this. Okay, so you should understand that too. Okay, so why I have drawn like that, okay, in coronal dentin and radicular dentin. So this is very important. So the curvatures of the dental tubule is more in a coronal dentin and less in the radicular dentin. Similarly, another point is here, okay, to understand is that, so it is a dentino-enamel junction, but this is a, as this is a cementum. Okay, so this is present in a root. Okay, so this is a dentino-enamel junction, this is a dentino-cementum junction. Okay, so it is not scalloped as that of the dentino-enamel junction. So you should remember this. So dentino-enamel junction, as I have discussed in an enamel chapter, there I have told that this dentino-enamel junction is in scalloped pattern with the convexity. This is a convexity is towards the dentin, and the concavity is towards the enamel. So you should remember this. Okay, so you can just understand by this picture that too. Similarly, as I have shown here, this is a right angle. Okay, so dentinal tubules make right angle, or it is perpendicular towards the pulp, towards the pulpal side with the pulp. In the enamel side, it is also at a 90 degree, or it is perpendicular to the enamel at the enamel junction. Okay, so it is very important. Another point you should understand from this picture is that, okay, so this is a first curvature. Okay, when we move from this pulpal towards the pulpal side towards the enamel side, okay, so this is the first curvature of the dental tubules, and this first curvature is directed towards the root apex. Okay, so this is a apical side of the root. Okay, so this is a root side, and this is a coronal side. Upper side is a coronal side, and another curvature is directing towards the crown. So this you should remember. Okay, so first curvature is directed towards the root, and second curvature is directed towards the crown. Okay, so just remember this. So the point I have told in this picture is that, first, there is a more curvature in the dentinal tubules of the coronal dentin than that of the radicular dentin. Another point that I have told here is that it makes the perpendicularity, okay, it maintains perpendicularity with the pulp and also the enamel on their respective side. After that, I have told about the dentino-enamel junction and the dentino-cementum junction. So dentino-enamel junction, it is scalloped, but dentino-cementum junction, it is not scalloped. And as I have discussed in the enamel chapter, it is scalloped with the concavity towards the enamel side and the convexity is towards the dentinal side, okay. And after that, I have told you that our first curvature of the dentinal tubule is directed towards the root and second curvature is directed towards the crown. So you should clear all your concept by this picture, okay. So both the pictures you can relate now. So in this picture I am showing the radicular dentin and here it is less curvature or the small curvature, okay. So it is just opposite of this, okay. Here is it is scalloped, here it is straight, okay. And this dotted line is a granular layer of the tomb, okay. So this is very important here, okay. So this is a distinguishing feature in the radicular dentin, okay. So at the junction of the cementum and the dentin in the root, okay, there is a dotted like structure, okay. Black dots are present there and that are called granular layer of the tombs, and I will be discussing with this granular layer of the tombs in detail in later in this video, okay, itself. So just understand those two pictures, okay. After that, I have shown you another two pictures, okay. So these two pictures are of exception, okay. So what I mean to say is here is that, okay, so at the incisal or the cuspal edge or at the root apex, okay, so incisal or the cuspal edge, the dentinal tubules are the straight, okay. So you should remember, okay. So if it is tooth, okay, so if it is tooth, okay, okay, so this is a enamel, sorry, this is a dentino enamel junction, okay. So this is a dentino enamel junction, just think, and this is a pulp, okay, junction between the dentin and the pulp. So at the cuspal region, okay, so the cuspal region or the incisal region, so at this region, okay, so as I have drawn here straight, okay, so there is a straight dentinal tubules present at the incisal or the cuspal region, okay. So incisal region or the cuspal region, it is present straight. Similarly in this granular layer of the tombs, similarly in this radicular dentin, okay, we can see dental tubule straight here too, okay. So it is also straight in the root apex side, okay. So you should remember those 2 points, okay. So all the dentinal tubules that are present in a tooth, okay, that is of S-shaped, okay, it is of S-shaped, but there are 2 exceptions. One exception is at incisal or the cuspal edge where the dentinal tubules are straight and they are perpendicular here also, and here also they are perpendicular. So don't get confused, okay. As you can think that, okay, dental tubule as they are S-shaped, okay, so they do not make a 90-degree with the pulp or enamel at their respective side, but at the incisor and the cuspal edge as they are straight, okay, so this would make the 90-degree. So don't think that like, okay, so don't think that like. So just you should understand is that at every condition, okay, so every condition they makes the 90-degree with the pulp or the enamel, but the difference is that at the incisor or the cuspal edge or at the root apex, okay, so the dentinal tubules are straight, they are straight, not resembling like that of the coronal dentin and the radicular dentin, okay. So as I have discussed with this figure a lot, okay, so I think you have understand this. After that, so I have covered this, okay. So primary curvature I have discussed. Similarly, okay, so we have curvature, 2 types of curvature we have. So I have prepared the proper note, okay. So when you will study all those notes before your exam, it will be very helpful, okay. So just after watching this video you can just go through the notes, okay. So the curvature, we have primary curvature as I have discussed in that picture. Similarly in secondary curvatures, okay, so secondary curvatures are the small curvatures. So what they are is, okay, so I will show you by the blue pen, okay. So what happens is that, okay, so when the dentinal tubules move from the pulpal side towards the cementum side, so during its path, okay, during its path along its length, okay, so there is small curvature present, okay. So there is small curvature present along the dentinal tubules, okay. So small curvature is present. Similarly here in this coronal dentin too, okay, so smaller curvatures are present there, okay. So those curvatures that are present along the length, okay, and they are sinusoidal in shape, okay. So they are sinusoidal in shape and they are along the length of the dentinal tubule. So those type of curvature are called secondary curvature, okay. So they are present over the length of the tubule and minute curvature or sinusoidal in the shape, okay. So now in this point, as I have written here, the primary curvature are formed due to the crowding of and the path followed by the odontoblast as it moves towards the pulp during the development, and from it moves from the more area towards the lesser area. So this point is very conceptual, you should understand it, okay. I will dissect this concept, okay. So what it means to say is, okay, so I will draw here, just think, okay. So this is a tooth, okay, and this is a dentino-enamel junction, and below that there is a dentin, and this is a pulp. So this is a pulp, okay. Okay, so just this is a enamel, this is a dentin, and this is a pulp. So now just think, as I have written here, okay, so from more area to the less area. So when as I have told earlier, odontoblast and the ameloblast, okay, so as I have shown here, so this is a ameloblast, okay, and this is a odontoblast. So ameloblast and odontoblast at the formation, okay, so at the beginning of the formation of the tooth during the odontogenesis they are closer to each other, but as the development proceeds the odontoblast moves towards the pulpal side. So just think like that, okay. So think this at the dentino-enamel junction, okay, so the area of this dentino-enamel junction and the area, okay, the surface area of this pulp, okay, it is less here, and the surface area that is present at the dentino-enamel junction is larger. So just think, okay, so if, okay, so they are present like that, okay, so they are the odontoblastic cells, okay, so they are odontoblast, okay, so they are present at the dentino-enamel junction by keeping a lot more space between them, okay, but when they will move towards the, okay, but when they will move towards the pulpal side from the dentino-enamel junction, they should have to be closer, okay, because there is an area less, okay, as the area is less than that in comparison to that of the dentino-enamel junction. So due to the crowding of the odontoblast, okay, when it is proceeding towards the pulpal side, that makes the curvature as shaped, okay, so that results in the formation of the primary curvature. So this is very important to understand. After that, as I have written here, it is less pronounced at the incision and the cuspal area, so I have discussed here in this figure. So now in another point, along its course it gives the lateral branches at every 1 micrometer. So this is very important data, okay, so 1 micrometer distance, which helps in a communication with the adjacent tubules or blindly inside the intertubular dentin. It may contain the odontoblastic processes. So this lateral branches means what, okay, so I will explain by this picture. So in this picture, as you can see, okay, so these black lines are the dentin, and these black lines are giving the lateral branches, okay, so small lateral branches, okay, so it is similar like that of the root, okay, so root of the plants, okay, so plant do have similar lateral branches, okay, so these are the branches, okay, these are the branches that are laterally to the main dentinal tubule, okay, so those branches are called lateral branches, and they are about at every 1 micrometer distance, okay, so if this is a dentinal tubule, so it gives lateral branch at this point, okay, and if it gives the lateral branch at this point, so distance between this is 1 micrometer, okay, so you can remember this. After that, the function of this lateral branch is to help in communication, okay, so it helps in communication with the adjacent tubules or blindly inside the intertubular dentin. It may contain the odontoblastic processes, okay, odontoblastic processes may be present in those lateral branches. After that, I have written here, tubules are tapered structure, that is T is to T, okay, so this is mnemonic here, so you can remember this, T is to T, okay, so larger towards the pulpal side, okay, so what it means to say is that, okay, so I will explain this. Okay, so tubules are tapered structure, so I will draw a small picture, okay, so dentinal tubules are like that, okay, okay, so it is like this, okay, so this is a dentinal tubule, okay, so that I have written here, tubules are tapered structure larger towards the pulp, so it is larger towards the pulpal side and it is smaller towards the dentino-enamel junction side, okay, so the enamel side it is a less tapered, it is more tapered at the pulpal side. After that, the ratio of the number of tubules per unit area near the pulp and the outer surface of the dentin is 4:1, so you should remember this, okay, so the ratio of the number of tubules, okay, so as we moves towards the pulpal side, okay, so as we during the, this is also relating with the clinicals, okay, so in clinicals when you move towards the pulpal side, the sensitivity increases, okay, because the number of tubules per unit area increases near the pulp, okay, but it is less at the enamel side, okay, so outer surface of dentin it is just 1 and it is 4:1, okay, the ratio is 4:1, so you should relate with these clinicals. Okay, after that, when the dentinal tubules are cut transversely, tubules appear circular with a dark spot at the middle and periodontoblastic space surrounding it. Dentinal fluid or the lymph are present in that and other constituents, okay, so I will explain this by a picture. So I have picture for this, okay, so this is a picture, okay, so in this picture, just for explaining this point, okay, so I will say you, so just think this is a transverse section. Okay, so we are cutting the dentin in a transverse section, so it appears like this. Okay, so it appears like this. Okay, so this is a dentinal tubule. If you consider this is a dentinal tubule, this is odontoblastic process. Okay, so this green line, okay, so this green dot, okay, so this green dot is the odontoblastic process present here, and this is a peritubular dentin. I will be discussing with this peritubular dentin, but properly this all thing that is present here is the transverse section of dentin, and it is a dentinal tubule. So odontoblastic process is present in a dentinal tubule, and the space that is left here, okay, so this space is a periodontoblastic space. Okay, so that I have written here. So in periodontoblastic space there is a presence of the dentinal fluid or the lymph. Okay, and so this is the fluid that causes the hypersensitivity in the tooth. Okay, dentinal fluids are the lymph, and it runs in those periodontoblastic space and cause the hypersensitivity. I will be discussing with this dentinal fluid and the lymph and the theories of this hypersensitivity sensitivity in upcoming videos. Okay, but for today, just you need to know is that, okay, so dentinal fluid or the lymph is present in the periodontoblastic space. Okay, and this periodontoblastic space, this dentinal fluid contain the collagen fibrils, plasma protein, glycoproteins, and the electrolytes are present in this dentinal fluid. Okay, we will be discussing with this dentinal fluid in detail in upcoming videos. Okay, so for now, just you remember those points: collagen fibrils, plasma protein, glycoproteins are present in this dentinal fluid or the lymph. Okay, and dark spot that is at the middle, that is a odontoblastic process. Okay, so I have picture for this also, another picture for this. So this is a transverse section. Okay, so this is a transverse section of the dentin. So as you can see here, this is a circular dentinal tubules, and the dark spot that are present in the dentinal tubules are the odontoblastic process. So now, okay, we have covered this. After that, the dentinal tubules are the longer than the thickness of the dentin, that is 3 to 10 mm buccal. Okay, so as I have discussed in enamel chapter, okay, so you should relate those both points. Okay, so in enamel chapter I have told that enamel rods, okay, or the enamel prisms, they are the length of the enamel rods or the enamel prisms are more than the thickness of the enamel. Similar like that to here in dentin, dental tubules are the longer than the thickness of the dentin. Okay, which normally is 3 to 10 mm. Okay, so the thickness of the dentin, which is 3 to 10 mm normally, generally. Okay, and among that too, it is higher at the buccal surface than after in the lingual surface, and in mesial and the distal surface there is a similar thickness of the dentin. Okay, so you should remember this point. Okay, so in buccal surface there is a more thickness, there is a presence of the more dentin, so there is a more thickened dentin present in the buccal side, and in the lingual side it is smaller than that of the buccal side, and similar thickness of the dentin is present in the mesial and the distal. Okay, and which both are smaller than that of the lingual surface. Okay, so after covering this, now our another subtopic of this dentinal tubule is a lamina limitans. Okay, so this is also very important here. So in lamina limitans, okay, so in lamina limitans is a calcified tubule wall, has inner organic lining that is rich in a glycosamine glycans, that is GAGs, which is similar to the lining of the lacunae in the bone and the cartilage. So lacunae are present in the bones and the cartilage. Okay, you have studied in your general anatomy. So this lamina limitans is a organic lining. Okay, I will show you by the another pen. Okay, so this is a organic lining. Okay, so this is a organic lining, okay, that lines the inner wall. Okay, inner wall of the calcified tubule. Okay, so if this is a dentinal tubule, okay, so the inner lining, so this is the inner lining of this dentinal tubule, so that at the inner lining this organic lining is present, that is called lamina limitans, so which is rich in a glycosamine glycans, and which is resembling with the lining of the lacunae in the bone and the cartilage. The important for the regulations and the inhibitions of the calcification of the dental tubule. So you should remember this. After that, some people think no such lining is present in the tubule. Okay, only if present plasma membrane blockage. So lamina limitans is just, you should remember is that, okay, lamina limitans is a organic lining that is present inside the, okay, inner surface of the calcified tubule wall. Okay, tubule wall, inner surface of the calcified tubule wall, that is rich in a glycosamine glycan, and that is important for the regulation and the inhibition of the calcification of the dentinal tubule. So as the age, okay, so as the age increases, okay, with the age there is a calcification of the dental tubules and there is a formation of the sclerotic dentin. I will be discussing this sclerotic dentin in this video too, but in later time. But for now, just you remember that lamina limitans is important for the regulation and the inhibition of the calcification. And also I have found this point is that, okay, many of the people thinks that there is no any such lining present at the inner surface of the calcified tubule wall. If present, okay, if present, then it is just like that of the plasma membrane. Okay, so plasma membrane is the covering of the every cell. Okay, so the plasma membrane is present. Okay, so some of group of the investigators, okay, some group of people thinks like that. Okay, so in cover this, after that in the next subtopic, so we have predentin. Okay, so predentin, it is a 2 to 6 micrometer in white, okay, and adjacent to the pulp, and it is pale staining. So it is pale staining, bit pale staining, and it is layer of the unmineralized matrix, okay, and composed of the collagenous and non-collagenous protein. As a matrix undergo mineralization at the dentin predentin junction, predentin becomes the dentin and new layer of the predentin is formed. Okay, so the thickness remains constant. Okay, so this point is very important. So this predentin, sometimes they can ask you in a viva. So actually predentin means, okay, dentin that is formed. Okay, so the proper adult dentin, okay, so it is a child dentin, we can say. Okay, so it is a child dentin. So every dentin that is formed in a tooth should go through this predentin. Okay, so what I mean to say is that, okay, so just think this is a dentin lamina junction, not tooth surface. Okay, so this is a dentin lamina junction and this is a pulp. Okay, and this is a predentin layer. Okay, so this is a predentin, so this is a predentin, and here there is a mental dentin. Okay, here is a mental dentin. Okay, so mental dentin is bit wider than that of the predentin. So okay, just think that, so at the pulpal side there is a presence of the predentin. So predentin is just unmineralized matrix, so there is no any kind of mineralization, but in dentin there is a mineral. So just that is the difference between the predentin and dentin. So this is very important. So what is the difference between dentin and predentin? So it is a just a matrix, but dentin, it is a matrix plus mineralized matrix. Okay, okay, so it is a mineralized matrix dentin. It contains just collagenous and non-collagenous protein, and it is present near the pulp. Okay, but dentin is present all over the, okay, all over this, between all over the part of the tooth between the dentinal junction and the pulp and this predentin. Okay, so as they are matrix, okay, so when this predentin, when there is a moving of odontoblast towards the pulpal side, okay, when there is growing of the dentin, so this predentin change into the, so this predentin change into dentin. Okay, so predentin just change into dentin and new predentin is formed. Okay, so new layer of predentin formed. So the thickness of predentin is always constant. If the predentin changes into the dentin, I mean to say is that if the predentin changes into the dentin, the new predentin layer is formed. Okay, so the new layer of the unmineralized matrix is formed, that is of 2 to 6 micrometer wide. So it is always constant, you should remember that. So the thickness is always constant, and I have written the difference between the mantle dentin and the predentin. So mantle dentin is a first formed dentin adjacent to the dentinal junction, and it is located adjacent to the pulp tissue. And mantle dentin, it is a 20 micrometer and it is 2 to 6 micrometers, so it is a bit thinner than that of the mantle dentin. After that, fibers are perpendicular to the dentinal junction here too, and fibers are in mixed orientation. Okay, so the collagen fibers, okay, so the fibers means collagen fibers that are present in the predentin are in mixed orientation. Okay, so they are in random orientation. Larger collagen fibrils are present that are called bone corpse fiber, and they are silver stained. Okay, so as I have discussed in my previous video, when I have discussed with the difference between mantle dentin and the circumpulpal dentin, there I have discussed that there is a presence of the bone corpse fiber in a mantle dentin, and here there is a smaller collagen fibrils present. Okay, after that the globular mineralization takes place in a mantle dentin, and it is a unmineralized dentin. Okay, so it is very easy to understand. And circumpulpal dentin, you can just recall. Okay, so there is a globular plus linear type of the mineralization. Okay, after completion of this predentin, so we have a peritubular dentin and intertubular dentin. Okay, so now in this peritubular dentin, okay, so they are the tubules that are, okay, so tubules are delimited by the collar of more highly calcified matrix called the peritubular dentin. Okay, so just that I have discussed earlier. Okay, so peritubular dentin is just a calcified like a mass. Okay, so it is a calcified matrix, mineralized matrix that just limits the dentinal tubule. Okay, so it just forms the walls of the, so it forms the wall of the dentinal tubules. It is a calcified matrix that forms the collar or the wall of the dentin. Okay, dentinal tubule, sorry. So it forms the wall or boundary of the tubule, and it is 9% more mineralized, okay, than the intertubular dentin. So intertubular dentin is the dentin that is present between the dentinal tubules. After that, it is less organic constituent is present there, collagen fibrils, and more sulfated proteoglycans is present there. After that, it is lost in decalcified section. So this is very important. Okay, so okay, this is very important, it relates with the clinical. So it is lost in the decalcified section. Okay, so when we prepare the decalcified section at those section, okay, we can't see this peritubular dentin. So it is lost in the decalcified section, but this intertubular dentin, it is present. Okay, it doesn't lost in the decalcified section. I will be showing you, but where it is. Okay, okay, I will discuss with this in this intertubular dentin, but just to remember, here it is lost and here it is gained. Okay, so it is not lost in the decalcified section. After that, it differs in the matrix, crystal arrangement, and the appears same. Okay, so here in the peritubular dentin, as I have told there, okay, it is twice thick in outer dentin and then in the inner dentin. Okay, so in this picture, the thickness of the dental tubule is greater. Okay, in this outer dentin, and this is smaller in the, and the thickness is less in the inner dentin. Okay, so okay, I have made mistake here. Okay, so this is, if it is towards the dental enamel junction, okay, so it can't be that much thick. Okay, so I have made mistake here. Okay, so sorry for that. So if it is toward, it should be towards the pulp. Okay, so it is just opposite. I have just made opposite. Okay, so it is dental enamel junction or dentinal cementum junction. Okay, just so I was thinking that. Okay, so just think this. Okay, so if, okay, so the dentinal tubule is, and the dental tubule is larger towards the pulpal end, but it is smaller towards the dentino-enamel junction. Okay, so it is the outer dentin and this is the inner dentin. So the peritubular, okay, so the peritubular dentin should be, okay, it is just a matter of common sense that, okay, so the peritubular dentin should be smaller. Okay, should be less thicker in this portion. Okay, so it should be less thicker in this portion, but it should be more thick. Okay, it should be more thick. Okay, it should be more thick towards the dentino enamel junction side. So it is very easy. So I have just made mistake here, I have just recalled, I have just corrected this now. So just you don't get confused. Okay, so sorry for that. So 0.4 micrometer is towards the inner dentin and it is towards the pulp, and 0.75 micrometer is towards the enamel side, dentin enamel side, and it is a, and it is of the larger thickness. Okay, so now in intertubular dentin, it is the dentin that is present between the dentinal tubules. Okay, so dentinal tubule is delimited by the peritubular dentin, and the intertubular dentin is present between the peritubular dentins, okay, or the dentinal tubules. So it is the main body of the dentin. Okay, it forms the space between them. Okay, so it is located between the zones of the peritubular dentin. It is less mineralized, half of the volume is of organic matrix, and it remains after the decalcification. So as I have written here, it remains after decalcification, but it is lost after the decalcified section. Okay, so after decalcification, peritubular dentin is lost because it contains inorganic constituent more. Okay, after that, I have written here, collagen fibrils are arranged randomly in a plane at roughly right angle to the dentinal tubule. Okay, and it represents the primary formative product of the odontoblast. Okay, it just represents the primary, okay, formative products of the odontoblast. So odontoblast may have formed this primary, it is a primary formative product, okay, of the intertubular dentin. Okay, so I have one picture to explain this intertubular dentin. So in this picture, as you can see here, okay, so in this picture I wanted to show you. Okay, so I have shown this picture previously, but those brown and these blue dots are not present in the first part of the dentin, but I have done little modification in this picture to show you the peridentin. Okay, so peritubular dentin is this brownish color. Okay, so this brown color is a peritubular dentin, and this green color line is a dentinal tubule. Okay, so it is a lumen of the dentinal tubule. This whole is a dentinal tubule, but it is a green color portion that is present here. It is a lumen of the dentinal tubule, and in the lumen there is a presence of the blue line. Okay, so if you can see or not, okay, so it will be clear. Okay, so this blue line I have drawn here. Okay, so blue line are present in this lumen of the dentinal tubule. Okay, so those blue lines are the odontoblastic processes, and the dentin that is present between the dentinal tubule is the intertubular dentin here. Okay, and this is odontoblast is present here, and this is a primary and secondary dentin, and this is a tertiary dentin. Okay, so dentinal lumen. Okay, so it is just lumen, dentinal tubule. Okay, so it is a wider here. Okay, and lot more space is present between them. There is sparse. Okay, so lot more space is present between them, and also random arrangement of the dentinal tubule is present there. Okay, so we have covered the peritubular dentin, intertubular dentin. Okay, after that, our next subtopic of this today's uh, video. Okay, it is a odontoblast and its process. So odontoblast, the diameter is 7 micrometer and the length is 40 micrometer. It is present at the junction of the dentin and the pulp. Okay, so it is present at the junctions, at the tight junction or the desmosomal junction are present in between them. Okay, after that lifespan of it is equal to the age of the tooth and do not undergo further differentiation. Okay, so lifespan. Okay, so the odontoblast one, it's once it is differentiated, okay, so it doesn't undergo further differentiation. Okay, and it is equal to the age of the tooth. So when the age dies, when the tooth dies, sorry, when the tooth dies, okay, so at that time odontoblast also die. After that, odontoblastic process composed of the microtubules and they run longitudinally through the tubules. Okay, so odontoblastic processes, okay, so they are constituted of the microtubules. Intermediate filaments are present in those microtubules and they run longitudinally throughout the tubule. Okay, processes sometimes extend to the enamel called the enamel spindle, and about this enamel spindle we have discussed in detail in the enamel chapter. Okay, so you can visit that. After that, in incremental line, okay, so in incremental line, so it is also called incremental line of the von Ebner's, okay, or the von Ebner's incremental line. Okay, we can tell anything. After that, it run perpendicular to the tubules. Okay, so if it is tubule, okay, so I will show you the picture. Okay, so after looking the picture you will be clear. So in this picture, as you can see here, so this black color, okay, so this black color that is running from the pulpal side towards the dentinal side, so they are the dentinal tubules. Okay, and those lines, okay, so those lines that are running like this, okay, so these lines are the contour lines of, sorry, they are the incremental line of the dentin or the incremental line of the von Ebner's. Okay, and the contour line that I was telling, okay, so contour lines are the accentuated line of the incremental line. Okay, so I have written here. Okay, so just to go through those notes, so it is representing the rhythmic deposition. Okay, so as we have discussed in the enamel chapter also, so there was the incremental line present there, incremental line of the radius. Okay, and the accentuated incremental line of the radius was there. Okay, and that is similar with that of the contour line of Owen's. Okay, so that is, so just I mean to say is that, okay, so incremental line of radius, okay, incremental line of the radius is similar with that of the contour line of Owen's. Okay, contour line of Owen. Okay, and the cross striations. Okay, so cross striations that are present. Okay, so cross striations are similar with that of the incremental line here. Okay, just they are analog of each other. Okay, so von Ebner's line, von Ebner's, okay, von Ebner's line. Okay, so von Ebner's line are analog with that of the cross striations, but this incremental line of the radius, okay, that is present in the enamel, that is accentuated, that is similar with that of the analog with that of the contour lines of the Owen of the dentin. Okay, Owen of dentin. Okay, so just you should remember this. After that, 4 to 6 micron apart in a crown, closer in the root. Okay, 5D increment is of 20 micrometer thickness. Okay, after that represents the growth pattern of the dentin. Okay, rhythmic deposition, growth pattern of the dentin, sometimes due to the disturbance in either matrix deposition or the mineralization, this incremental line may become the accentuated called the contour line of the Owens, as I have discussed here. Okay, so a contour line of the Owens intercept the dentinal junction and meet accompanying stria of radius that was formed at the same time. So this is very important. Okay, so this came from this. Okay, so contour line of Owens of the dentin and the incremental line of the radius, they both are the analogous. Okay, and if they are formed at the same time, they accompany each other by crossing the dentin enamel junction. Similarly here, the cross striation are analog with that of the von Ebner's line. Okay, incremental lines. After that, Owen described, okay, result from the coincidence of the secondary curvature between the neighboring dentinal tubules. Okay, okay, so originally, okay, so as I have written here, okay, so there is a contour line of Owen formation. Okay, so it is formed due to the matrix deposition or the mineralization disturbance. Okay, so disturbance is present in the matrix deposition or the mineralization, but also Owen. Okay, so the name contour line of Owen came from the Owen. Okay, so the Owen described, okay, it as a line that is resulting from the coincidence of the secondary curvature. So as I have discussed earlier, secondary curvatures are the sigmoid like curvatures that are present along the length of the dentinal tubule. So they are formed by the coincidence. Okay, so they are formed by the coincidence of the secondary curvature between the neighboring dentinal tubules. After that, neonatal line. Okay, so it is similar with that of the enamel. So neonatal line, if it is present in the dentin, then it is a dentinal neonatal line, and if it is present in the enamel, then enamel neonatal line. Okay, so it is just you should remember here is that, okay, so neonatal line that is present in dentin is mostly in the deciduous teeth. Okay, so deciduous dentition. Okay, so and the first permanent molars. Okay, deciduous teeth and the first permanent molar. After that, in interglobular dentin. Okay, so I'm discussing with this interglobular dentin, so it is very important. Okay, interglobular dentin, it is the area of the unmineralized or the hypomineralized dentin where the globular zones of the mineralization calcospheroids have failed to fuse into the homogeneous mass. Okay, so what happens here is that, okay, so just think, okay, after the matrix deposition there should be mineralization. Everybody know this. Okay, so there is a one globular pattern of the mineralization. Okay, so what happens in a globular pattern of mineralization? Okay, so the globules, okay, so the globules of the minerals, okay, so these are called calcospheroids. So each circle, okay, so this circle is called calcospheroids. Okay, and they are the globules of the minerals, so they fuses and they form the mineralized mass, okay, over the matrix. So when these globules failed to fuse with each other, okay, so just think, okay, so now another this globule, okay, so this 4 number globule is it, okay, and it is unable to fuse with this 3 number globule. Okay, so there is a formation of this space here. Okay, so this space, okay, that is formed between this globule, okay, that space will be unmineralized and just will contain the matrix. Okay, and after when we see this in a light, okay, so they appears the dark. Okay, so those unmineralized area of the dentin are called the interglobular dentin, and they are rich in a sulfur. Okay, so they are high in the sulfur and area of unmineralized hypomineralized dentin. Okay, it is prevalent, so this is important here, it is prevalent in a vitamin D deficiency or exposure to the high level of the fluoride at the time of the dentin formation. Okay, so when the dentin is forming, if the person suffers from the vitamin D deficiency or it is exposed to the high level of the fluoride, so at that time there is a high chance of the formation of the interglobular dentin. After that, just below the mental dentin where the pattern of the mineralization is largely globular. Okay, so in mental dentin there is a globular mineralization. Okay, so below the this mental dentin, just below the mental dentin, where the pattern is just a globular. Okay, so there is a high chance of the formation of the interglobular dentin. After that normal architectural pattern of the tubules remain unchanged because it defect is the, okay, so as I have told you, okay, it is just a defect of the mineralization, not the defect of the matrix formation. Okay, so during the matrix formation there is a formation of the dentinal tubule, so the normal architectural patterns of the dentinal tubule is unchanged. Okay, so you should remember this. Okay, so the change is just in the mineralization, the architectural pattern is similar. After that, no peritubular dentin exists here. Okay, so no peritubular dentin is here, and the order, okay, so in crown, in cervical portion it is more, and after cervical it is more in a middle portion, less in the middle portion, sorry, so the more in the cervical portion, then after in the middle, then after in the intercostal areas. Okay, similarly in the root, in cervical there is more and in the middle there is less. Okay, so just to remember is that you should remember this order. Okay, so interglobular dentin is maximum present at the cervical portion, either in the root or the crown. So I have the picture. Okay, so this is a picture of the interglobular dentin. So in this picture, as you can see, it is like just like a stars. Okay, and they are dark. Okay, so when we see in a microscope they appear dark. Okay, and as you can see, these are closer to the mental dentin, just beneath the mental dentin. Okay, mental dentin is that dentin that is near the dentinal junction, and it is just beneath the mental dentin, and this is appearing as a dark star. Okay, so they are the interglobular dentin that is rich in a sulfur. Okay, high in a sulfur content. After this is our last paper of today's video. So in this paper, okay, so granular layer of the tomes. Okay, so in granular layer of the tomes, okay, so I will show you the picture first of all, then after I will discuss with the notes. So in a granular layer of the tomes or the tomes granular layer means the dotted layer. Okay, so this is a dotted layer here. Okay, that is present between this is cementum and this is a dentin, so it is present between the dentino-cementum junction. Okay, so there is a predentine too. Okay, so after the predentin, okay, this is a predentin, and after that there is a presence of this tomes layer of the tomes granular layer, okay, towards the dentinal side. Okay, so just remember cementum, predentin, tomes granular layer, and this is a dentinal tubules. Okay, so in radicular portion of the teeth, peripheral border of the mental dentin adjacent to the cementum has a granular appearance, and this granular appearance layer is called granular layer of the toms, and it is increasing in a granularity occurs from the cemento enamel junction towards the apex of the root. So this is important here. So when you move from the cemento enamel junction, okay, so cementum enamel junction, okay, so I will see you in the picture. Okay, so just think, so this is a cementum, okay, so this is cementum, okay, so okay, so this is a root, okay, and this is a cementum, and this is a junction, okay, so this is a CEJ. So when you move from the cemento enamel junction towards the root side, okay, root apex side, so the granularity increases, okay, and it is highly granular at the apex of the root. And this point here it is written, it is suggested that it represents the section that is made through the looped terminal, okay, so it is formed, it is made through the sections, okay, it represents the section that is made through the loop terminal portions of the dentinal tubules, okay, so dentinal tubule just form the looping at that portion, okay, and that gives the representation of the granular layer, okay, so dotted layer is formed there due to the looping of the dentinal tubule. But in recent interpretation, okay, but in the recent findings, okay, there we can see that, okay, this layer contains the special arrangement of the collagen and non-collagenous matrix protein at the interface between the, okay, so previously it was think that, okay, Tom's granular layer was formed by the looping of the terminal portion of the dentinal tubule, but after recent findings, okay, we can say that it is formed due to the special arrangements of the collagen and the non-collagenous matrix protein at that point, that is between the dentin and the cementum. Now our another topic is the sclerotic dentin or the translucent dentin. So sclerotic dentin is the tube, okay, dentin, okay, it is just, you should remember in sclerotic dentin is that, okay, the dentinal tubules that is present in this dentin are translucent, okay, so when we see by the microscope they are translucent. It is because that the dentinal tubules, okay, so the periodontoblastic space that is present in the dentinal tubule, okay, it is occupied by the calcium, so calcium occludes that space and it causes the translucency, okay, so also it is called translucent dentin. So tubules get occluded with the calcified materials and with the age glassy appearance and they becomes translucent, okay, so they appears like the glass, okay, increases with the age and glassy appearance is present there, and it can be physiologically or the pathological. So okay, it can be physiologically by aging, okay, so when there is increase in age, okay, so when the age of any person increases, okay, then there is a increase in a number of this sclerotic dentin, okay, so the dental tubules are calcified. But in a pathological condition like that of the artesian and the caries, okay, so in slowly progressing caries and this artesian, okay, slowly progressing caries, okay, slowly progressing caries, okay, so in the case of the artesian and in the slowly progressing caries there is a formation of the sclerotic dentin. Also I have written here, okay, slowly progressing caries resistant to the caries. So sclerotic dentin is a type of adaptation here, okay, in slowly progressing caries it is sort of the adaptation that resists the caries formation, okay, so it just wanted to stop the caries to that particular position. After that, most commonly found in the apical third of the root and in the crown midway between the dentinal junction and surface of the pulp, okay. After that it reduces the permeability and prolonged pulp vitality, okay, it reduces the permeability so that it resists the caries, okay. After that, in the most important point here, okay, in sclerotic dentin sometimes they can ask in a viva, okay, so this sclerotic dentin have its own function in the forensic odontology, okay, so in forensic odontology this sclerotic dentin is one of the criteria for the age determination, and the method of determining the age by using this sclerotic dentin is called Gustafson's method, okay, so Gustafson's discovered this, so it is called Gustafson's method, okay. After that I will be discussing the dead tracts, okay, and dead tracts is the last topic of today's video. So in a dead tracts it is characterized by the degenerated odontoblastic process, okay, so it is just, so sclerotic dentin is just a precursor of the dead tract, okay, so in dead tract it is totally dead, okay, so there is a deposition of the calcium, odontoblastic processes also are dead, degenerated in dead tract. But in sclerotic dentin just there is a calcified material present in the periodontoblastic space, okay, so periodontoblastic space is just occluded but odontoblastic processes are the vital, okay, it entraps the air, appears the black in a transmitted and the white in a reflected light. So this is important here, okay, so it appears the black in a transmitted and white in a reflected light, so you should remember this, okay. So in this second point how you can remember is that, okay, so you can remember by, okay, so this BT, okay, so black means transmitted light, so between, okay, so black transmitted light, okay, so black is present in a transmitted light and in white it is reflected light, so white WR. Okay, so you can remember BTWR. So BTW means between, so between R. Okay, so you can remember this, between R. So this is important here. This is mnemonic. You can just make your own mnemonic to remember this, but I have made like that. Okay, so BTW and R. Okay, so between R, between R's. Okay, so black in transmitted, white in reflected light. After that, may occur in the teeth containing the vital pulp as a result of the caries, obtrusion, abrasion, and cavity preparation or the erosion. Okay, so as I have told here, okay, in sclerotic dentin there is a prolonged pulp vitality. Okay, and here also it may be, okay, but it may be present in the teeth that is containing the pulp vitality as a result of the caries. Okay, so sclerotic dentin is just a precursor of the dead tracts. Okay, so when the caries are extreme, okay, so the sclerotic dentin is a type of adaptation and it fails to control that caries, and when the caries is just larger and it is growing, okay, so there is a formation of the dead tract odontoblastic processes, okay, which is live here. Okay, that is not degenerated in a sclerotic dentin, but it is degenerated in a dead tracts. Okay, so it is just dead. Okay, so the name suggests us that the dead tract means it is totally dead. Now it may also develop in the region of the cusp or the incisal edge due to the death of the odontoblast occurring as a result of the overcrowding. Okay, so at the incisal and the cuspal region, okay, so there is a overcrowding of the odontoblast and that can cause the death of the odontoblast and odontoblastic process simultaneously. So there is a muscle formation of the dead tracts towards the cuspal region and the incisal edge. Okay, and there is a presence of the reparative dentin at the pulpal end of the dead tracts. Okay, so there is a presence of the reparative dentin at the pulpal end of the dead tracts. Okay, so I have picture of this dead tract. Okay, so here is a dead tract. Okay, so as I have told here in this paper, so as I have told there, so dead tracts are mostly found in the cuspal region. Okay, cuspal and incisal region, because there is overcrowding of the odontoblast and then that can cause the death of the odontoblast and odontoblastic processes simultaneously. So that dead tract is formed here. Okay, and also another thing that I have told here, okay, it is presence of the reparative dentin at the pulpal end. Okay, so dead tract. Okay, so as I have discussed in my first video, one we have reparative dentin and another we have reactionary dentin. So in reparative dentin there is a death of the odontoblast. Okay, and as I have discussed here, okay, so odontoblast, okay, due to the death of the odontoblast. Okay, so as there is a presence of the death of the odontoblast, so new odontoblasts are formed and they form the reparative dentin at the pulpal end. Okay, so you can just understand like that. So we have completed all the topics of today's video. Okay, and I think I have clear all your concept in these different topics of dentin. So if you love this video, please do like, subscribe and share with your batchmate. Till then, stay happy, stay healthy and stay tuned. Okay, so I will upload the part 3 video of the dentin very soon.